Hello, welcome to lecture 13 of Pharmacognosy and Metabolic Engineering NPTEL online certification course. So, in lecture 13, we will be dealing with the engineering tropane alkaloid pathways in plants. So, in the previous lecture, I have discussed about the tropane alkaloids, their biosynthetic route, their early information available, and with the advanced research, the latest information about the pathway available, where tropane alkaloids are localized in the root, which part of the root, which tissues. So, these all aspects I have discussed in the previous class. Now, what we will discuss is this: based on the knowledge what we have acquired in the previous class, can we really make application? That means, can we really make transgenic plants to produce these alkaloids in higher amount. So, which is basically the job of metabolic engineering. That means, we will be manipulating or augmenting the metabolic pathway either by expressing a gene or by blocking a gene, and as a result of this there will be changes in the metabolite profile, and from there it will be possible to assess whether the strategy followed for metabolic engineering has been successful or not. To start with, so we will talk about transgenic Atropa belladonna expressing H6H gene, because what I have said in the last class is this, that H6H is the rate limiting step, and we will see what will happen when H6 gene is expressed in Atropa belladonna, and then we will also talk about the co-transform hairy root culture for enhanced tropane alkaloid production. We will discuss few more case studies of the pathway genes, and then we will also discuss the more latest case studies where the newly discovered enzymatic route, the genes for the newly discovered enzymes were inserted or overexpressed in the plant, and as a result of that what is going to happen. So, this I have already mentioned that this is the basic route of tropane alkaloid, and the product of our interest are hyoscyamine, scopolamine, calistagine and so on. And these are the some of the plants I have mentioned this is henbane or Hyoscyamus niger, and this is Atropa belladonna, this is Dhatura stramonium and so on. So, to transfer the desired gene into the target plant, one has to use the proper vehicle. So, the most common one is agrobacterium mediated transformation, agrobacterium tumefaciens mediated transformation, that will discuss about, we will not discuss about the protocol, but we will discuss the outcome of such transformation to make transgenic plants which express the H6H gene. The other one is this hairy root. So, in that context we will discuss the co-transform hairy roots. So, now I will start with the metabolic engineering of medicinal plant transgenic Atropa belladonna with improved alkaloid content. Okay. First of all, so the target plant is and the source plant. Source plant means that the source of the gene. So, that let us define first. The source plant is Hyoscyamus niger, and from where the sorry, go back to the previous slide, from where the H6H gene has been isolated, that is the gene source, and the target plant is Atropa belladonna. I have written on the top. Now, question is this, that why Atropa belladonna has been chosen. Okay. So, that we need to understand. To answer this, the first important point is this, that Atropa belladonna is a hyoscyamin rich plant, and there is almost no scopolamine available in the plant, or no scopolamine detected in the plant. That is the point, right. So, if H6H gene is used for transferring if H6H gene is used to transfer, or other word H6H gene can be transferred into the Atropa belladonna by using Agrobacterium tumefaciens mediated transformation. So, where this along with that plant selectable marker should be used, and that should also contain a promoter and a terminator. So, this is a vector to be constructed in this way. So, this is the right border, this is the left border, and this will go inside the Agrobacterium A tumefaciens, and so it has, this is the binary vector what I have discussed in the previous class, which contains your gene of interest along with the plant selectable marker, and both are kept inside the left border and the right border. In addition to that, there will be another plasmid which is there in the Agrobacterium, which is again a disarmed plasmid, which contains the virulence. Just I am briefly revising, help you to revise the previous class. So, this is another plasmid, and so this virulence will act in trans and allow this tDNA containing the H6H gene to go out and integrate into the Atropa belladonna plant. Okay. So, Atropa belladonna was chosen because it is hyoscyamin rich plant. So, let me briefly once again remind you the enzymatic reaction. So, enzymatic reaction is like this, that it converts hyoscyamin, I just denote with H, to 6 beta hydroxy hyoscyamin HH, and these two S scopolamine. And the first one, if you remember that I have also mentioned that this is basically the hydroxylation, and this is the epoxidation. And what I have also mentioned that these constitute 95% of H6H activity, and this constitute 5% of the H6H activity. Now, the beauty of this experimental design is this: Atropa belladonna is a hyoscyamin rich plant and there is no scopolamine. So, therefore, if you transfer the H6H gene into Atropa belladonna, and if the gene function, then what will happen? The plant is hyoscyamin rich, that means the transgenic plant should be able to should be able to utilize the available substrate. Utilize substrate. What is the substrate? Which is H, very efficiently. Now, in the background there is no product, product is scopolamine, that is not there. So, the result, if you get a positive result, then that you can interpret that is because of the H6H expression. If there is, if the plant produces scopolamine and if you transfer H6H and if you get the enhancement, then you cannot be sure that enhancement is because due to a natural mutation available there, or it is a line which already producing more scopolamine which you have not checked before. So, then it is difficult to interpret the result, but where here it is rather relatively easy, because there is no product. So, once you transfer the gene, it will make the, if it makes the scopolamine, then that scopolamine is because of the transgene HCH expression, and that is why it happens. Fine. Now, let us see the result, what has come to the, what is the result. Go to the next slide. So, the result is very clear. The transgenic Atropa belladonna produces scopolamine as the principal alkaloid. You can say almost it is exclusively scopolamine. Okay. So, that means normal Atropa belladonna plant contains hyoscyamin. So, if you have a control plant and if you have a transgenic, so transgenic means it has H6H. So, in control plant what you find is this, it contains hyoscyamin, but in transgenic plants you got S. So, almost no H. So, that means all H got converted into S. Fine, very exciting results, but again question came to the mind of the scientist talking about relating this to right. Okay, just a moment. Scientist, by raising that issue once again, one is the hydroxylation, another is the epoxidation. What I have said that H converted into HH and then HH converted into S, and as this constitute 95% and this constitute 5%. Now, question is this, that what is the role of epoxidation here? Okay, the H6H is non-existent in Atropa belladonna, you have transferred the gene. So, H6H successfully transferred the hyoscyamin to 6 beta hydroxyhyoscyamin and then to scopolamine. Now, the question is this, that is 5% epoxidation is that much efficient to convert the huge amount of hydroxyhyoscyamin to scopolamine, because what I have said that it is almost scopolamine is the exclusive alkaloid. So, that means there is also negligible trace of hydroxyhyoscyamin, that is the intermediate. So, that is only possible if the epoxidation is very efficient, but epoxidation is only 5%. So, that there lies the confusion. So, to clear the confusion, what scientists they did, actually they raised tobacco plants. One is wild type, another is transgenic. So, the transgenic one I put it in this color. Transgenic means these are the tobacco where H6H, sorry H6, expressed lines and this is the wild type. Now, you put them in a solution okay. So, here what is there in the solution basically here you will do some feeding. So, feeding what you feed? You feed here hyoscyamin, you feed here hydroxyhyoscyamin, you feed here hyoscyamin, you feed here hydroxyhyoscyamin. Now, remember we are talking about tobacco or Nicotiana tabacum. I just use the scientific name, that is better. So, the Nicotiana tabacum you have made the construct H6H you have expressed. So, tobacco plant is not producing this alkaloid. Okay. So, it is basically producing a nicotine alkaloids, right. Now, this is a wild type plant. So, wild type plant means there is no gene here. If you feed the hyoscyamin, what is going to happen? Plant will absorb hyoscyamin and after a period of incubation, if you analyze, what you will find here is this, that here you will find upon analysis it is only hyoscyamin. Similarly, this one upon analysis you will feed only hydroxyhyoscyamin. Now, if you take the H6H express lines, so here there is also no substrate, but the gene is there and enzyme is functioning. So, here if you feed hyoscyamin and if your H6H is functioning, then what will happen? Here you can see HH as well as scopolamine, if both are functioning, and when you are feeding HH. So, if epoxidase is sufficient 5% epoxidase is sufficient enough, then you will see only scopolamine, and exactly that is what happened. Wild type what you fed, you got the same thing. In the transgenic tobacco plant, when you fed the intermediate when you fed the first substrate, that is hyoscyamin, which is converted into hydroxyhyoscyamin and scopolamine. So, you can get both HH and S, but when you fed hydroxyhyoscyamin, all hydroxyhyoscyamin got converted into scopolamine. That means what? The 5% epoxidase is sufficient enough to convert the huge amount of all hydroxyhyoscyamin into scopolamine. Okay. So, this is a beautiful experiment scientists have designed, and what they have also found that these tropane alkaloids synthesize in the root and they are transported into the aerial part. So, this is now the in summary, the gene source was from Hyoscyamus niger, the gene was transferred into, this is Hyoscyamus niger plant and this is Atropa belladonna. The gene was transferred into Atropa belladonna because it is a hyoscyamin rich plant, and scopolamine was almost the only alkaloid present in the aerial part of transgenic belladonna, and we have confirmed that indeed 5% epoxidase is sufficient enough to convert the huge amount of hydroxyhyosamine into scopolamine, and this was the first metabolic engineering paper with tropane alkaloid, and that published in 1992. And I am teaching this subject for 20 years. I got a lot of advanced papers I read, but I find this is a brilliant paper, and perhaps the best paper to teach first metabolic engineering class, because this is a very positive result with the metabolic engineering application. And remember that at that such an early time is 1992, this is 2023. So, even in that time it was so successful. So, now we will talk about the production of tropane alkaloid in genetically engineered root cultures. Fine, Atropa belladonna plant is producing, but what about to produce this tropane alkaloid in root cultures? The advantage of root culture is that you can grow the root culture all around the year, and therefore, it is always better to have the root culture, and that too if you use hairy root, it is always better. So, the construct what I have drawn briefly, I am not drawing the promoter and terminator here with the because of the time concern. So, please for one time I will put it so that you will not get confused. P T, here is a P and it is the T. So, this is the selectable plant selectable marker, and this is the gene of interest. Okay. So, this construct what I have shown in the previous previous slide, that this was transferred into Atropa belladonna plant using Agrobacterium rhizogenes. Now, we will transfer this into the hairy root. Now, hairy root is basically formed, I have mentioned in the previous class very briefly perhaps, that this is because of the RI plasmid. So, Agrobacterium rhizogenes has the RI plasmid. So, that means it has the region of virulence as well. Along with that, it has the main region of tDNA. That is the tDNA region, where you have the genes for hairy root formation. Here you have the genes for hairy root formation. Those are mostly the Rol genes and the genes for auxin synthesis. Now, this RI plasmid, what happens that as a result of Agrobacterium rhizogenes mediated transformation, this tDNA is transferred into the target plant tissue, and as a result of that what happens, because this contains the gene of auxin synthesis, as a result of that hairy roots are emerged from the site of infection as a normal one. Now, the difference what you should remember, that here we want to have the hairy root phenotype. So, hairy root phenotype must be maintained, whereas in case of Agrobacterium tumefaciens mediated transformation, we use disarm strain. So, there will be no crown gall, but here hairy root phenotype must be maintained. That means, we cannot disturb the wild type tDNA. So, now this let us see, this is the Agrobacterium rhizogenes. So, it has its RI plasmid, what I have drawn here in the right side. So, RI plasmid contains the its own tDNA, right border and red border, and I said this contains the several genes, for example, the genes for auxin synthesis, and also there are several Rol genes present, which will determine the phenotype of the hairy root. Okay. In addition to that, what I have said here that it also contains the virulence genes. So, this virulence is present outside the tDNA and this virulence will allow the this tDNA to go out. Now, in addition to this, here we are putting the synthetic plasmid. This is the synthetic plasmid in a binary vector. So, this synthetic plasmid contains the synthetic tDNA having the right border and the left border, and this particular construct is inserted here. That means, here you have the H6H and plant selectable marker. So, this is the binary vector, okay. B, or you can say B19, B19. Okay. Now, what will happen? This here is no vir, but this vir allows its own tDNA to go out. This vir also allow this binary vector's tDNA to go out as well. So, as a result, what will happen? You will see the, I will just draw it here. You will see the, you call co-transformed hairy roots, co-transformed hairy root culture can be developed, which is basically coming from two tDNA. One is its own tDNA which originates from RI plasmid, and the another one is basically the binary vector tDNA which contains the H6H. So, what is happening here is this, that so this tDNA, the left one, this is responsible for hairy root phenotype and this is responsible for the H6H gene in the hairy root. So, the first tDNA will go containing the auxin genes, it makes the hairy root. Second tDNA also will go along with that, and that will become a part, and eventually you have to select it the correct hairy root line which contains the H6H. So, that is possible because in this selectable marker you have put a kanamycin resistance gene. So, the root can be grown in that kanamycin, and hairy roots are normally very sensitive to kanamycin, but if kanamycin gene is inserted there, then it is possible with a low dose of kanamycin hairy root can survive, and from there co-transformed hairy roots can be recovered, and these hairy roots in principle have the H6H gene, and because it is expressed under the control of promoter. So, it will express, and as a result of that it will make the product. So, now let us see what has happened as a result of this outcome, that this is hyoscyamin 6 beta hydroxylase gene, that is H6H, and this gene was placed under the control of a 35S promoter and a NOS terminator, because it is a, what I said in the previous class, it is a constitutive promoter, and they have used a binary vector system. That is why I said that without disturbing the RI is there, but you put this insert, this you can insert it inside, and there are different ways to put this vector inside the wild type agrobacterium. One is electroporation, another is called triparental matting, but we cannot discuss that, but that is a separate concept. So, here mostly we are dealing with the pathways and engineering. So, we will not talk about those tools and techniques more here, and then the engineered belladonna hairy roots contain up to 5-fold higher concentration of scopolamine than wild type hairy root. That is interesting, okay, because wild type hairy root normally you will find mostly hyoscyamin, because Atropa belladonna hairy root does not make scopolamine, but this one is making scopolamine because of this H6H gene, and the content of this also increased, and they said that such genetically engineered hairy roots should be useful for enhancing scopolamine productivity. So, that is there, and but yeah, okay. So, I will not go to further complicacy, because they have developed these hairy roots in a different way. One way of developing hairy root is that they have the transgenic Atropa belladonna plant expressing HCH. So, that plant they have infected with the wild type agrobacterium rhizogenes. As a result of that, the hairy root which produced, apparently it appears like a wild type, but it contains the H6H there. So, that is why such hairy root also produces scopolamine. So, that is why in some lines scopolamine is also detected, in which came from the line generated from the transgenic plant, but the one which I have developed, I have just discussed about the co-transformation. This is much a clean way, and this way whatever scopolamine is formed, that is because of the gene which is inserted and makes the hairy root, and hairy root contains H6H. So, now subsequently scientists what they have done, they have used another plant as a model, which is Hyoscyamus niger itself, and Hyoscyamus niger also known to produce hyoscyamin. Now, they are putting PMT and H6H either as a single drive or jointly. So, what they said that the normal wild type root contains it's only if the content of scopolamine is only 43, whereas wild type Hyoscyamus niger the normal the scopolamine content is 43 milligram per liter, whereas when the single H6H gene was inserted, because of this gene expression it produces 184, and when you have both PMT and H6H, the level reached up to 411 milligram per liter. So, that means the huge increase when you compare with the wild type. So, here is the pathway PMT, here is the H6H. So, one of they have made the construct in this way, that the one of the earlier pathway gene, one is the late pathway gene. So, earlier if it expressed, it will push the pathway in this direction, and the late one will push this direction. As a result of this resultant activity, you will get more product accumulation. So, another work what has been done with Atropa belladonna, this time they have used expressed tropinone reductase 1 and tropinone reductase 2, and tropinone reductase 1, as I have said, that contributes to scopolamine root, and tropinone reductase 2 that contributes to calistagine. So, true tropinone reductase is basically this is the branching point enzyme, and root culture strong, which shows strong expression, it shows the products either tropin or pseudotropin. Okay. So, when TUR1 is expressed, it will produce the tropin which can be detected. When TR2 is expressed, they found that it is pseudotropin is detected, and when is high pseudotropin is detected, as a result of that they found that higher level of calistagine. Okay. And when they have expressed TR1, so that leads to the formation of hyoscyamin and scopolamine, 3-fold more hyoscyamin and 5-fold more scopolamine compared to the control line. And in this case, what happens when the scopolamine and the hyoscyamin contents are more? So, what will happen? Automatically the calistagine content, automatically is not the right word, the calistagine content they found is reduced. Why it is reduced? Because this is basically they are competing for the common substrate tropinone. So, when tropinone 1 is active, more active, so the flow is this way, therefore, it limits the tropinone availability for the TR2, and that is why you get higher activity of this, but lower activity of this. So, this one is a very successful paper that was also published in Journal of Experimental Botany long back, and the this one probably with this I will end the class because of the time constraint. So, the other thing I will discuss in the next class. So, here what is this? The functional expression of tropinone reductase 1 and hyoscyamin 6 beta hydroxylase from Hyoscyamus niger to tobacco. That means, the source plant the source plant is Hyoscyamus niger and your target plant is basically the Nicotiana tabacum. So, again tobacco plant is not producing tropane alkaloid. So, they have transferred TR1 as well as H6H in the tobacco and they wanted to see what has happened. So, here the interesting thing is this, that tobacco plant express TR1 and H6H, but it will not produce the tropane alkaloid because the substrate is not available. But if you do the feeding of tropinone and feeding of hyoscyamin, that leads to the expected reaction product. That means, if you put feeding to tropinone and hyoscyamin, then this is TR1. So, this will work and then you will see the formation of these products. Okay. But interesting point is this has produced this level in higher amount, that is no problem, expected product has produced, but in addition to that what they have also noticed that it produces some unexpected substances under transgenic background. So, the plant is meant for producing nicotine. So, that background is wild type plant biochemical background is now changed because you have inserted 2 genes for tropane biosynthesis. So, it is some sort of perturbed or disturbed, and as a result of that they found unexpected substances like acetylated form of tropine. So, this is tropinone to tropine. So, what they found? They found acetylated form of tropine that was detected, and the leaves of transgenic plants it showed higher content of nicotine than control plant. That is also very interesting because these leaves produces more higher content of nicotine, and they said the nicotine level is up to 13-fold higher when compared to the wild type. That means, under a transgenic background for tropane alkaloids, it somehow boost the nicotine formation. In addition, nicotine related compounds such as anabasine or nicotine, bipyridine, anabasine, these alkaloids were identified in the transgenic plants, which are normally not identified or not detected at that level in the wild type plant. But in the transgenic tobacco, when it expresses H6H and TR1, it is, although it is meant for the tropane alkaloid, but unusual changes occurred leading to formation of as a result of unexpected change leading to formation of this minor nicotine alkaloid, which is now detectable in the transgenic plants. So, that is very interesting. So, inserting transgenic plant can change the background of the normal biochemical metabolism. As a result of that, you can get some unexpected products, and this paper published in 2002. So, I will end the class here with this. Thank you very much.